right, well, I think the numbers of the people joining have sort of stabilized. So I just wanted to welcome everyone to this evening's conversation. We're lucky enough to be joined here this evening with the leaders of the UC San Diego Return to Learn program. And I'm just so grateful that they were willing to give their time to come and have a little bit more of a technical conversation about the plan to open up the campus. And I'm really excited about the progress that they've made recently. And I'm just so grateful that everyone was able to join us this evening. So tonight we'll be hearing from Chip Schooley, who's a professor in the Department of Medicine and the Division of Infectious Disease and Global Public Health, and the Faculty Director of Global Education and the Senior Director of International Initiatives in the Division of Infectious Disease and Public Health, and the co-director of the Center for Innovative Phage Applications and Therapeutics, and the key lead of the Return to Learn program. We'll also be hearing from Angela Scioscia, who's the Interim Executive Director of the Student Health and Wellness Initiative, and from Dean Cheryl Anderson, who's the Professor and Dean of the new Herbert Wertheim School of Public Health and Human Longevity Science at UC San Diego. We also will be joined by Professor Natasha Martin, who's an Associate Professor of Medicine, and she'll be telling us about her modeling work, and by Professor Rob Knight, who's a professor in the Departments of Pediatrics and Computer Science and Engineering, and the Director of the Center for Microbiome Innovation, and the co-director of the new Excite Clinical Testing Lab. So please join me in welcoming our guests. This is truly an all-star panel, and I am just so grateful for your time. And so today we will be holding the questions until the end, and then Scott Rifkin and I and Quinn Wynn will be moderating the Q&A session at the end. We will be using the questions that were submitted in advance and questions that you type in during the chat. So without further ado, I'll turn it over to Dr. Schooley.
Thanks very much, Kit. It's great to be with everyone today, and I'm glad that we could all get together today to talk about some of the aspects of the planning process. The main focus today is going to be some of the mathematical modeling that Natasha Martin, Ravi Goyal, Victor DiGurtolo, and others have been doing. It's really formed a framework around which we've thought about a lot of the issues that we need to respond to, react to, and monitor as we try to get things up and going to a greater extent here at UC San Diego. Tonight, I'm just going to give a very brief overview of the program. There is a town hall for faculty on Friday in which much more detail about other aspects of the program will be talked about, including Chris Longhurst's first presentation of a very innovative approach to use Bluetooth proximity features of cell phones to help with some of the contact and case notifications. So tune in Friday as well to hear a broader overview of some of the issues that will be touched by all the modeling we'll be talking about today. After a brief overview, Angela Scioscia, who is the Interim Director of the Student Health Service, is going to be talking about the efforts to keep tabs on both the viral activity within our student body and student interactions with the overall Return to Learn program. Dr. Cheryl Anderson is leading the public health intervention component of the program and has set up a team to interact with all people that are found to be shedding the virus through the surveillance program or who have been exposed and require isolation or quarantine. She'll tell you a bit about the approach there. Robin Knight has been working around the clock on both a on-campus high throughput laboratory that will substantially increase our testing capacity here on campus, and on a comprehensive proposal that went into the NIH yesterday to augment that. So if he's not comatose, he'll join us and talk a bit about some of the testing activities that have been added to our program. If he isn't able to get here, I can talk a bit about it as well. And then we'll move to the open colloquium that Kit and Quinn and Scott will chair. Rob is here. Good. He said he has joined us, although he is comatose. So this would be a good chance to really interact with him in his native state. Okay, so let's go to the next slide. And one of the things I want to emphasize is that we've tried in this overall program to build in a substantial amount of overlap and resilience, in the sense that we are hoping to be able to monitor and respond to the viral activity in multiple dimensions. The first part of the program, of course, is risk mitigation, and this has to do these days mainly with face coverings, masking and physical distancing. Those two things, as Bob Redfield of the CDC said today, if every American would wear a mask for 6 to 12 weeks, this would be over. And that is really the mainstay of a lot of what we're going to be focusing on throughout the entire process. A lot of work has been done to reconfigure some of the places that people interact with each other with plexiglass and face-to-face interactions. A lot of work has been done with thinking about cleaning and sanitation. One thing that has happened, though, is as the epidemic has matured and we've learned more about transmission, we're much less worried about fomite transmission than we were back in April and May, when you were afraid to go to the grocery store and peel an avocado. So things have really changed now, and the emphasis now really is on airborne spread. We have a very ambitious program to detect virus in multiple ways along the campus. We are going to be looking for people who have symptoms and very quickly get them tested so that we can make sure they get the health care they need, get them into isolation, and identify people that might have been in contact with. We have a very aggressive asymptomatic testing program that we are standing up with Rob's lab leading the charge after a tremendous contribution over the course of the summer by David Proud in the Calm Lab, and that will still serve as a backup to the program as we move forward. Environmental monitoring is primarily at the outset wastewater monitoring, and Rob will talk a bit about that at the end, because we've already got this up and going and have already had a very successful dry run, I guess I should say wet run, that gave us an opportunity to see how it all worked. And then we're working closely with the County of San Diego to look at the context of the epidemic in the county as we are part of the county, and what goes on there is going to go on the campus and vice versa. So we're very closely working with the county health department, and we'll be comparing and integrating our data with theirs. Intervention includes case isolation and contact tracing that Dr. Anderson will talk about, exposure notification, both by the contact tracing teams and by the app that you'll hear about from Chris Longhurst on Friday. And as we get going, molecular sequencing that will allow us to identify case clusters as we move forward. All of this is interdependent, all of it is adaptive, and we're going to learn as we go and flex up and down different parts of this when we learn which parts are most effective, and as we react to the evolution of the epidemic in San Diego and on the campus. So let me turn now to Dr. Scioscia, who's going to talk to you a bit about the approach to viral detection and the screening program, both from symptoms and from the perspective of the virus. Angela?
Yeah, thanks, Chip. Good evening. And so I'll be focusing a lot about some of the new things that relate to students particularly. But you know, the daily screening has been around for a while. It began with the employees, and we introduced this to the entire student body, and they are starting to sign up, and we even have them doing it before they arrive on campus, using the same tools of if they have exposure, we're getting them tested, as well as if they have symptoms. And in those two settings, we're having them stay where they are in place and not come onto the campus, or if they're residing on campus, to stay in their bedroom until they can be properly evaluated. If they have none of those things going on, they get the green thumb, just like the rest of us. So then the other thing is, you talked about the waste sampling, but I think the next phase now is the asymptomatic surveillance process, or screening tools that we're going to be using. For the faculty and staff, this is a voluntary process. For the students, though, we have decided that there will be mandatory twice a month. You may say, why are we doing something a little differently for the students? It's really because of the significant rate of asymptomatic disease in this younger age group. That and the fact that they had a higher positivity. So taking those two factors into account, we felt that testing, surveillance screening for the student body should be done at a more frequent basis. This applies to all students that are coming onto campus, and it can be for a variety of reasons. It could be as an athlete, it could be because they're a resident, or a class, or a social interaction, or they're using the rec facilities. So they will be into this program. We want them twice a month, separated by no less than 12 days and no more than 16 days. We also have a number of student populations that we're trying to do some baseline testing, and I'm going to speak quite a bit about the undergraduate process because this is a very large group of students moving in. But for the others, we've had baseline testing for the students in the graduate and professional area that have been living on campus. They also have been doing the screening and then can get supplemental testing. And for any of the graduate students who arrived after August 12th, they had a test within 48 hours of arrival. We began large-scale testing for off-campus students this week. Actually, it's last week, and we have them coming to a specific area on the campus. We're using the parking lot. You may have driven by it and wondered what's going on there off of Torrey Pines, the 102 lot, and we're doing large-scale testing for our student body. We have a number of sites for testing, just to remind folks. We're in partnership with the health system, and the health system has them distributed throughout San Diego. But then on the campus, we opened up the Price Center, which is staffed by student health staff. Our nurses and nurse practitioners and physicians and MAs are all there, but we're supporting not just the students, but also campus employees. So you can actually have your testing. You can even get a flu shot at the Price Center. It's a nice, convenient location on campus. This parking lot location is for large-scale testing, though, and is a unique thing that we just began and will be only open on the month of September, essentially. So I want to talk a little bit more on the next slide, if we could move to that, about the undergraduates. I think we're all a little apprehensive because we're bringing in a large number of new individuals to our campus. Many of them are coming to a college environment for the first time in their life. They're leaving their families. So we, in response to the activities we saw on other campuses around the country with some significant outbreaks, actually looked at our original plan, which was originally a test on arrival, and decided actually to implement a more thoughtful, I think, and detailed plan, which involves more testing. So I'll walk you through that. So these individuals will be arriving. They start coming this Saturday and will arrive over a period of 10 days. They come in at anywhere from around 900 a day to 600 a day each day, and it's a very steady move-in process so that when they move into the suites, it's de-densified and it's slow moving, lots. You know, in the past we had many families and students. No, this will be very controlled. Each student has only 2 support individuals. When they arrive at the parking lot, our testing area, they will get a PCR-based test. They will then, after that test is done, they will move to the other parking lot on the side, and then they will be directed to their actual location on campus. They'll have 45 minutes to unload their belongings. They will all, on arrival actually into the parking lot, be assured that they are masking. They will be checked for symptoms, any exposure, any COVID history, etc. That will occur in the parking lot on arrival. They will be instructed, and the students have been told this in messages very clearly, that they will be expected to mask, have a face covering on campus at all times, including when they are in their residential units. In this period of time, they will be wearing their masks in what we call phase 1, everywhere except when they're in their personal bedroom. Remember, they are only 1 student to a bedroom, or when they're in the shower, of course, or need to wash their face or shave. Otherwise, they are to be masked at all times in their residential unit. Remember, they're coming in over a period of time, so some of the residential units will have everybody in on day 6, others will have everybody in on day 8, 9, or 10. After a period of 12 days, when everybody has been in the unit for a total of at least 12 days, which gives us a wide window of exposure, they will have another test. If that second test is negative, then at that point they will have been on campus a period of basically 2 weeks, at least, some longer, with 2 negative tests. At that point, they can move to phase 2, in which they are not required to wear a mask within their residential unit. They also can't visit students in other units. The minute they come out of their unit, they are to mask. They mask in the hallways, they mask everywhere, even in phase 2. The only place that they basically on campus are not wearing their mask is, of course, in the swimming pool, vigorous exercise like the rest of us, but for regular activities, they are masked except in their residential units in phase 2. In this process, though, we also are anybody who is positive will be moved to isolation. I'm going to talk about that. If someone were to develop symptoms in phase 1, they would also be tested. If positive, they would move to isolation as well. So this is the plan that we will be using over the next, we'll see it, 10 to 12 days as we bring these students on board. Again, they arrive starting on Saturday. Campus is going to feel a little different with these 6,700 individuals on our property. Next slide. And so one of the things we're doing is to make sure, should we have a student who is has contracted the virus, whether they're symptomatic or asymptomatic, they will be moving to isolation houses. If they are a resident of the campus, they will move to these specific buildings. When they're there in the isolation, they have their own bedroom, bathroom. They're the only individual in the apartment. They have food service by housing and dining direct delivery. The medical team from student health calls them every day. They're also giving counseling and psychological support from the CAPS team, as well as academic support. We, in collaboration, the student health is working with Dr. Anderson's team and the public health team to make sure the contact tracing and everything is going very smoothly. And similarly, exposed cases are giving quarantine support. They actually is quarantine housing option for us on campus as well. So now I'm going to turn over to Cheryl, and she'll speak a little more about the contact tracing and case investigation.
Yeah, thank you, Dr. Scioscia. This is a real treat for me to be with you all this evening. Thank you for having us. I want to talk a little bit more about how, when we have these early detection plans on the campus, how we plan to interrupt the transmission of SARS-CoV-2 infection. So here you see that a person receives a positive test, and our goal is to, within 24 hours, get a hold of that individual so that they know that they have tested positive. When we talk with that person, one of the key things that we're trying to do is to not just understand what their signs and symptoms might be, whether or not they're at high risk for developing a serious case of the disease, whether or not they may be at high risk for transmitting disease, but to how many people they're likely to transmit this to. So again, we want to interrupt that process. So we talk with the person who's infected, and we ask them about their demographics, basic health information. We really want to get a handle on, you know, their work situation, their social situation, their recreational situation, what kinds of community activities they're engaged in. I characterize that as where you live, learn, work, play, and pray. In the midst of getting all of that information, we also try to understand what's next for them and what barriers might be associated with these subsequent actions. What's next is that they need to self-isolate. This is a part of what we think of as that getting rid of the possibility of inadvertently shedding this virus across the campus and across our community. If there are barriers to doing so, we have conversations about those, try to provide wraparound services. And they are also given a notice from the health department. And this notice allows them to be able to have intelligent and credible conversations with their work supervisors, et cetera, because often this means that they're not going to be in the workplace. And then we follow individuals for a 14-day period. The frequency of following them differs depending on what part of our activities we're in, but generally it's every other day for our campus. Now we know also who their contacts are, their close contacts. These are people within whom they have been less than 6 feet for more than 15 minutes. And we want to get a hold of those close contacts within 24 hours as well. And so this is 24 hours of them being named by the person who has the positive test result. And what we ask of the close contacts is very similar to what we ask of the case, with the exception of some expanded information around symptoms. We really want to understand, are they currently symptomatic, what their testing needs might be, that's largely driven by their symptoms, and giving instructions as to how to monitor those symptoms so that a testing protocol can be followed that is responsive. And then, as we did with the person who had the positive test result, we talk with them about their activities, how important it is to quarantine, which is now a 14-day period of being out of general activities within the community, any barriers that might exist. You can imagine people may have had the best of intentions to run errands or pick up various, supplies and things of that sort. And we facilitate wraparound services to ensure that we can get those things to happen without them being in the community setting. They also receive a notice from the health department, and they too are in conversation with us over the next 14-day period. So this really summarizes what we do around the contact tracing and with the goal of really interrupting the transmission. So now I'd like to pass things on to Dr. Natasha Martin and her team to talk a bit about our modeling process. As Dr. Schooley mentioned, a lot of the interventions that we came up with were based on the results of this modeling. So on to you, Dr. Martin.
Great. Thank you, Dr. Anderson. It's really nice to be here and talk about the modeling that we've done. I just want to say that I'm really presenting on behalf of a larger team, and this model was actually developed by Ravi, Goyal, and John Hotchkiss, who are at Mathematica. And Ravi is fortunately here to answer more technical questions about the model at the end of the presentation. But I'm really here to talk about how we've conceptualized it and what we're trying to learn from it. And I just wanted to start with really a word about modeling and where it's useful. I think one of the main points where modeling is useful is defining data which needs to be collected to reduce the uncertainty. There is a lot of uncertainty about what will happen in the fall, and I think the more data we collect, then the more we can refine our models and our according processes to really take an adaptive and considered and data-driven approach. So models are particularly useful in examining the relative impact of different policies. They may be less accurate at predicting the absolute impact or the absolute numbers of infections, predominantly due to uncertainty in parameters such as, for example, behaviors which haven't occurred yet. So what we've done here is really use modeling to try to understand the relative benefits of adjustments that we can do on campus to increase campus safety. And I want to really underscore that our process is iterative and adaptive. What I'll be showing you today is our first iteration of the model. We'll be collecting data, we'll be refining it, we'll be adapting both the model as well as our approach as we go, as we collect more data and learn. Thanks. So the preliminary modeling that we did examined what testing frequency we would have to do in order to achieve a high probability of detecting an outbreak when there were less than 10 detectable infections linked to that viral introduction on campus. Many of you may have heard about that or maybe seen the work that we did. That was really initial modeling. And what we've done subsequently is developed a network agent-based model of UCSD campus transmission. And this model is much more sophisticated. It allows us to look at the particular, campus interactions and campus dynamics that we have to assess the impact of various structural configurations and changes we can do on campus, such as changing housing policies, such as changing class configuration and having hybrid class instruction. It can also be used to answer questions about the impact of having prevalent infections at the start of a term or external persistent seeding of infections from the community as we expect to occur over the course of the term. We can also look at the impact of interventions such as isolation, contact tracing, and quarantine, which you just heard about, as well as the potential impact of behavior such as masking and social distancing. And finally, of course, we can also use it to explore the impact of asymptomatic testing frequency. And in particular, we looked at differences in terms of testing among residential students or those coming onto campus. So the model, as I mentioned, is an agent-based network model. So we simulate individuals and they interact on networks, which I'll talk about on the next slide. But broadly speaking, the model simulates SARS-CoV-2 transmission. And that part of it incorporates aspects of the natural history of disease, which is shown here on the right-hand side, where individuals can become infected, and if they're infected, they will progress through various latent and infectious periods, which are either asymptomatic or symptomatic. Individuals can be detected through diagnostic testing, again, either asymptomatic testing or symptomatic testing, and a small fraction of individuals may require hospitalization. So the population encompasses roughly 46,000 interacting individuals. That's comprised of 15,000 undergraduates who are residing on campus or coming onto campus. Additionally, we simulate 15,000 undergrads who are not resident and do not have in-person classes, because we still assume that they'll be interacting with these other students potentially in social situations. We additionally simulate 8,000 graduates as well as 8,000 faculty and staff who are working on campus. Now, importantly, we do incorporate outside infections. So we include a bolus of infections at the start of the term. We assume a 0.5% prevalence among returning individuals to campus. And we also include a sort of ongoing importation of infections from outside of the community. And we generate estimates and predictions of that incidence based on a separate suite of county-level forecasting models that we've developed. Now, the modeling that I'll show you today does not incorporate additional preventative measures of things like wastewater testing and digital exposure notification, which you've heard about, and which we're hoping to collect data and incorporate into the model as we learn more about them as we go. So we simulate PCR testing, which is 80% sensitive and 99.5% specific, with a 24-hour test turnaround from test to results. We simulate the entry testing protocol that you heard Angela discussing in terms of individuals being tested on return to campus, as well as the 2 tests for undergraduate residential students. And so that sort of testing protocols assumes that we would catch the majority, though certainly not all, of initial infections because the tests are not perfect. And so therefore we do start the simulation with a certain number of existing infections that have been not detected. We also additionally look at periodic testing of various frequencies, ranging from monthly down to twice weekly, among resident students or students coming onto campus. We assume that others, such as faculty and staff or students who do not have in-person courses, are tested monthly, even though they're recommended to be tested every other week. We simulate public health intervention, isolation of individuals on the day of a positive test receipt. We also incorporate contact tracing, which traces suite mates of that individual as well as classmates in close proximity, so along those networks, which I'll get to in a moment. And we simulate immediate quarantine of close contacts. And finally, we look at a scenario and the impact of masking and social distancing. And what I'll show you today is a simulation assuming that with masking and social distancing, there would be a 50% reduction in transmission. So here is a schematic of how the networks are constructed. And this particular figure shows you the on-campus students. So these networks are based on actual housing and class registration data that we've received. And so this schematic shows you, for example, for an individual student here in red, they may be placed in a room with 1 or more roommates. Now, in practicality in the fall, we know that individuals will not have any roommates, they'll live in singles, but we wanted to use the model to explore the impact of that and, if we had doubles or our full occupancy. Additionally, those rooms are situated in suites, which may have shared bathrooms and are within a broader residence where individuals are interacting. Students are also interacting within their classes, so students are assigned to classes based on the actual registration data that we saw last year with kind of regular registration as well as now, looking at the impact of classroom size caps as well as hybrid distance instruction. Now, those residential and classroom networks are static. They're fixed across the term, but we also assume that there is a network of interactions that occurs randomly on campus that changes every day, just as people kind of interact with each other on campus and bump into each other. Now, as I mentioned, the model also includes off-campus students and faculty and staff and their interactions, and it assumes that those off-campus individuals have a background risk of acquiring infection due to their residence in the community. And so here are the model results of how structural and behavioral changes on campus can impact the basic reproduction number, or R0, as you've heard it, on campus. So the first 3 bars on the left show the substantial reduction in R0, which is expected with the existing structural changes that have been proposed, that are implemented for this fall, including de-densification of residences and the implementation of hybrid instruction with class caps. So the green bar on the left shows doubles in in-person instruction, what we would expect with that simulation. The cream line shows doubles in in-person instruction with a class size cap. The yellow shows singles and hybrid instruction with class size cap. So what we are planning for in the fall is this yellow. And you can see that with those changes, we've dramatically reduced the R0 from an estimated, greater than 6 to 2.5. And the final bar emphasizes the critical importance of student behavior in terms of masking and social distancing, which can reduce the R0 to near or below 1, depending on the effectiveness and the compliance of that intervention. So this particular bar shows what we would expect to see if we assume a 50% reduction in transmission with these behaviors. Next slide. So this slide shows the average outbreak size associated with each viral introduction on campus. And here we can see, like in the previous slide, that the residential and class instructional changes substantially reduce the average outbreak size associated with each viral introduction by about 75% from the green column to that singles and hybrid instruction with class size caps in the yellow. Now, masking and social distancing shown in the last column could further reduce this average outbreak size to less than 2 infections associated with each viral introduction. So it has a huge impact. This behavior does have a substantial impact on reducing outbreak size and transmission. So this slide examines how asymptomatic testing frequency affects the size of outbreaks associated with each viral introduction. And I know this is a complex slide, so I'll walk you through it. On the left-hand side, you can see that with all of these are assuming our structural interventions as we have them, that single housing, hybrid instruction, as well as masking. And what we're varying here now is the asymptomatic testing frequency. And you can see that as we increase the testing frequency from monthly, shown in the blue column, all the way up to twice a week, 2 times a week, shown in the green column, the average outbreak size is actually low in all the scenarios, regardless of the testing strategy, at below 2. So on average, two infections for every viral introduction. But in the middle of this slide, what I've shown you here is the distribution of those outbreak sizes, because that's just the average on the left. So how are the outbreak sizes distributed? And you can see that the vast majority of outbreaks are small, less than 10 for each viral introduction, but there are a small number which can exceed 20 infections. And if you look on the right-hand side, here in the right-hand figure, I've shown you the maximum outbreak size. And so here we can see a greater relative benefit of increased testing in reducing the maximum outbreak size, with greater testing reducing the likelihood of very large outbreaks. But I would also point your attention to the big uncertainty bounds in these simulation figures. So there's high uncertainty in the maximum outbreak size due to stochasticity in the system, as well as, of course, there would be additional simulation, different scenarios if there were less masking compliance, for example. Next slide. So the previous slides looked at clusters of infections that were linked to each viral introduction. But as I mentioned, the model incorporates for this sort of ongoing persistent viral introduction from the community and multiple outbreaks happening at the same time. So what is the impact of the strategies we're talking about on the peak numbers of things we're looking at and the cumulative numbers across the term? Here on this slide, we've looked at the impact of testing frequency on peak isolation housing need. And what we've seen in this figure, as well as in other simulations, is that under a range of scenarios, the modeling predicts we're likely to remain within our isolation housing capacity for on-campus students, which is 606 beds. And that more frequent testing could lead to slightly more isolation housing need because we're detecting infections, but still well within our capacity. Now here we looked at the impact of testing frequency on the cumulative number of infections across the total 10-week term. Now we've split the infections by how many are occurring as incident infections that are imported from the outside community, and that's in yellow, and how many infections have occurred due to transmission occurring on campus, which is in the navy blue. So you can see that regardless of the testing scenario, these columns are showing different testing frequencies, again from monthly up to 2 times weekly. There's an important fraction of infections which are imported from the community which are unaffected by our testing, right? Those are the bars in yellow. More frequent testing reduces the fraction of transmission which is occurring on campus, so that blue contribution. And so we can reduce that campus level of transmission. One thing that I would say though is that it is uncertain kind of what actual number of cumulative infections we will expect to see at the end of the term. That's highly uncertain, dependent on many of things, one of which of those is student behavior. And finally, this slide examines the relative impact of using less sensitive tests. And so it indicates that for a given testing frequency, using a test with, for example, 10% less sensitivity, so 70% sensitive instead of 80% sensitive, really only has a marginal impact on our predicted cumulative number of infections at the end of the term. But it also shows that the benefits of testing more frequently outweigh the drawbacks of a less sensitive test. So overall, I think we've found that, masking and social distancing is clearly critically important to reducing the reproduction number and, the outbreaks that we see on campus. Regardless, the vast majority of outbreaks are likely to be small, kind of less than 20, and the average outbreak size is predicted to be low. But the maximum outbreak size it could be large, and it could be reduced by more frequent testing. Although again, there's uncertainty in that absolute number due to many things like stochasticity and behavior. Now, more frequent testing will reduce campus transmission, but again, a substantial fraction of imported community infections will remain. And high frequency testing with less sensitive tests, if they're available, such as potentially with antigen testing or other novel testing methods, are worth the loss in test sensitivity. Now, I want to go back to what I mentioned at the beginning, and that is that, the modeling can help us understand the relative benefits of these scenarios. It's not a crystal ball to tell us exactly what's going to happen in terms of number of infections in the fall. There is a lot of uncertainty in that. But the uncertainty in the model can help us understand what data we need to collect, and then refine the model based on the data that we have and adapt both the model as well as the approach. And so we're collecting data in terms of clustering, contact rates, adhering of masking, contact tracing, etc. And our process is going to be iterative. We'll collect the data, we'll refine the model, we'll adapt the projections, and we may update the approach. For example, if we see that there are clusters of infections among particular populations, we may adjust the testing frequency in accordance to that. Now, again, the model at the moment does not account for potential additional preventative benefits of our wastewater detection strategies for early detection, molecular epidemiology, digital exposure notification, etc. These are all things that we can incorporate into the model to help us understand the impact. And as we generate data on them, we'll be incorporating and layering them into the model as it's developed. And finally, I think one of the important things we want to do with this model is to both understand the impact of the strategies on campus, but also disseminate this information back to the students and the broader UCSD community to help guide behavior so they can see how the choices they're making are likely affecting what we're going to see on campus and our resulting approach. And now, and so just to sort of conclude in terms of what we're planning to do. So again, we want to take this data and we want to translate it into policy. So we'll be continually monitoring daily these data streams in terms of health data, in terms of epidemiology data. And we have an entire kind of matrix of things that we're monitoring, not just numbers, but trends over time, and try and understand how those trends are changing both within our campus as well as in the community. And we're monitoring these as we go, and we'll have a daily technical review of these data and these trends. We'll feed that up to the campus administrative leadership, and then we'll try to understand the significance of the trends in the data and the changes that we see, the context that these changes and trends are occurring, and what we can do on campus to affect them, and therefore the strategy that we take. And again, we want this to be a targeted, data-driven implementation approach. We want to make decisions that are based on the data that we see, and we'll refine those as we go and adapt them as we accumulate more information. And again, we're going to look at a wide range of responses depending on the origin, severity, and potential impact of the intervention. We do not want this to be a binary, we're open and we're closed approach. We'll be continually looking at multiple streams of data and adjusting our approach as we go with varying levels of intensity depending on what we see as we go. Okay. And so now I'd like to actually pass it over to my colleague, Dr. Rob Knight, who will talk about the testing strategies that he's developed, as well as discuss the wastewater monitoring that we're doing on campus. Both of these to detect our outbreaks early and to reduce the risk of viral transmission. Dr. Knight?
Great, thanks Dr. Martin. So, thanks Dean Pogliano for the invitation to participate in this and speak to the group. Looking forward to the Q&A. So the wastewater testing program is an integral part of Return to Learn, but we've started relatively late in that what we're doing currently is a pilot, although a pilot that we're rapidly scaling. So over the last month and a half, we needed to establish whether, like other cities, we could use wastewater to predict SARS-CoV-2 infections, and then we needed to scale it down to the individual building level, which had never been done at the time that we started. So what we've been doing is working with the authorities at Point Loma, where we've established that we have a few days lead time between when we detect viral RNA signal in the wastewater compared to caseloads for the whole city of San Diego. And we've also been working at two specific medical sites in the UCSD system, which I think I'm not allowed to say the names of when we're recording this, where the goal was we know the patient loads at those particular sites, and then we can correlate those patient loads with the wastewater signal from those particular buildings. The campus one was our negative control. So in retrospect, had we realized that that signal might turn from negative to positive, we might have placed it differently. And what the intention when we do this for Return to Learn is going to be is that to the extent possible, each wastewater sampler is going to be sampling from a manhole that drains one building, ideally a residential building or a complex of residential buildings, rather than the situation with this particular sampler where to get enough flow while campus is largely shut down, we needed to drain a fairly large catchment area of different buildings, which included NSB, which I think resulted in a lot of additional excitement and correspondence, given that many of you work there. What we're doing for the wastewater testing is we're amplifying 3 viral genes by qPCR. So we're reading out the CDC N1 and N2 primers and the WHO E primer pair for those of you who are aficionados of this sort of thing. And we're also using human RNase P as a control for specimen integrity, which works fine for wastewater specimens like it does for human specimens. What we found was basically every single day at the 2 medical sites, we got a detectable and positive signal, and this has continued for 6 weeks now. On the campus site, the signal was negative every single day until, predictably, the day before the Labor Day weekend, when it turned positive for the first time. And what you saw was within 2 hours of getting that positive signal, rerunning the PCR, to confirm that it was positive on new technical replicates of the same specimen. That escalated to an enormously effective campus response where hundreds of people were contacted and tested, and all of the students tested negative. Again, I think this is well known. 2 employees tested positive, and we think they were likely the source of that signal. So being able to test large numbers of people rapidly in response to one of these events is the capability that we've already demonstrated in the Return to Learn program at UCSD at this point, which is pretty impressive. What we're hoping is that next time there's a positive signal, we won't have to go through quite such heroic efforts. One really important problem is the way the wastewater is collected. You can do what's called a grab sample, which is literally you open up a manhole, you put a bucket down the manhole and you grab some of the material. But you can only do that at very specific times of day when the flow is high enough. And most times if you try to do a grab sample, because of water efficiency policies on campus, you won't find anything flowing there. So, what we do instead is we use 24-hour aggregate samplers, where we have a robotic device that samples from the manhole every 15 minutes to every hour, and that's flow weighted so that we get an overall aggregate sample. And that's been shown in many studies now to be much more accurate for SARS-CoV-2 prediction of clinical cases when doing the grab samples. The auto samples that do this are in nationally limited supply. So there are two main suppliers, YSI and Teledyne, and those two suppliers initially were able to get us 3 samplers, which is how we're doing the pilot we told you about. Another 5 arrived today, and we're working with Dr. Martin and her team to figure out how to place those optimally across campus. We have another few dozen arriving by the start of October. And what we're doing is initially focusing on housing, where the models are by far the most effective, given what we know about student activities and student life. Although there's very much an intention to be able to extend this to the research building. I understand there was some substantial concern about research that's going on in SARS-CoV-2 in NSB, Tata Hall, other research buildings, and could this have led to the signal? We think the answer is no for 2 reasons. So one is that we were able to identify positive cases in the buildings. The second is that we identified the signal from all 3 of the primer pairs that we looked at. And our understanding is, and this is confirmed by analysis of the BUAs that people have, our understanding is that most research going on is currently looking at single genes and does not cover the whole viral genome. So even if the virus were present in the lab and discarded, if you only have a plasmid that has 1 gene, we're not going to detect that anyway in a signal consistent with what we really observed, which picked up all 3 primer pairs. However, if you are doing research on SARS-CoV-2 and you have the full-length genome, or you have multiple genes in your construct, it's really important that you let EH&S know about that with their BUA, and really important that you let us know about it, because not only is it necessary for to meet federal requirements for biohazards research, which you should be doing anyway, right? But also it'll greatly decrease the chance that a positive signal in the wastewater due to lab activities causes a sort of panic with where we have to test hundreds of people at short notice, which is a lot of stress for the various lab teams. So we're rapidly scaling up the wastewater sampling capabilities. We're also looking at things like, could we manufacture more of the devices on campus under contract to Teledyne? So SIO already manufactures equipment under contract to Teledyne for various marine applications, for example. Or can we get the companies to scale up production or allocate us more? Or could we build them on campus? One issue is that wastewater monitoring is a regulated field, kind of like medical devices are regulated. And so buying a commercial unit, you comply with all of those regulations, which I didn't even know existed until a couple of weeks ago. Whereas if you build your own, getting all the compliance issues worked out is not exactly straightforward, kind of like with clinical devices and clinical testing. So that's the wastewater side of things. So far, there's one campus sampler. Very soon, we hope there are going to be a lot more, and we think that'll be really important for providing a complementary signal that both helps us inform the epidemiological modeling that you've heard about, and also lets us see, are there any populations of people on campus that we're missing with the clinical sampling. I was asked to say a little bit about the Excite Lab. So this is a collaboration between myself, Jean Yo, and Louise Laurent, and our labs, where what this is a new CLIA facility. It's run under the CLIA license of the Biochemical Genetics Lab, operated by Dr. Nyhan, which may be a familiar name to many of you. He's been on this campus for over 50 years, I believe. And so essentially supported by Retentalin, we have a very high capacity liquid handling infrastructure. Right now, we are EUA to do clinical testing on nasal swabs collected in either MTM or in Maui buffer, for those of you who are aficionados of this sort of thing. We are rapidly bridging that to other collection methods, including dry swabs. And there's a lot of interest in saliva. We're looking at setting up the saliva tests as rapidly as possible. And right before this, I was on a call with the Saliva Direct team at Yale, where we hope to be EUA for saliva by the end of the week. You might be wondering, so the nasal swabs, if you've had an MP swab, you know that that's extremely uncomfortable, and it's this huge swab about that long that scrapes your brain. The nasal swabs are not like that at all, so they're about that long. They only have to go about 1.5 centimeters into your nares, where you scrape the septum on both sides and then drop it into a tube and you're done. So it's the kind of thing that you can do yourself, and we're pursuing EUA to make it possible for people to do self-collection unobserved in their dorm rooms or at home, instead of doing it provider observed, which is what the EUA currently covers. Like I said, there's a lot of high capacity automation involved in this. We are guaranteeing return to learn that we can do 6,000 tests a day, although we certainly hope to scale beyond that. And what our limitation at the moment is staffing, where we're trying to hire a whole bunch of additional clinical lab scientists. So if you know any who are good at it and are either looking for work or might rather help campus test for SARS-CoV-2 than whatever they're doing currently, please encourage them to come our way and reply to our job ads. If you're wondering about how much saliva you need, and you've seen videos of people spitting and spitting to try to get 5 or 10 mils into a Falcon tube, we've also been doing a lot of assay optimization on that. So currently the assay requires 50 microliters of liquid saliva. Typically we're asking people to collect about 300 microliters of saliva, which is enough to repeat the assay several times and not call people in for a new sample. The platform that we're using to do it is we're collecting into these 1 mil matrix tubes, which, as you can see, come pre-labeled with a little barcode on the end. And what you can do, even today actually, is you can take the UCSD app on your phone and you can scan that barcode, and that uniquely associates it with your employee ID, or if you're a student, with your student ID, and then that connects to the university's EPIC system so that we can record your test result. So your next question is probably, that tube looks like a pretty small target. How do I actually get my spit in there? And to address that problem, we've turned to the very high-tech solution of the funnel. Although before you laugh, there's a lot of engineering that went into this funnel, and Rodolfo Salido, a very talented bioengineering student in my lab, his family is in Tijuana, so he's very excited about getting this testing strategy to work cross-border, designed this and 3D printed it. We worked with the design lab at QI to refine the design, and we're currently talking to injection molding manufacturers who can stamp millions of them out at a few cents a unit, which is what we're planning to do for Return to Learn. So the instructions are basically, you get the tube and you get the funnel, and you put the funnel on the tube, and you need to get some saliva in there. So basically you just spit into the funnel, and you'll notice the tube has a little slot on it. So if the liquid is up to the bottom of the slot, so it didn't quite get enough in there that try, if it's up to the bottom of the slot, you have enough. And as long as the froth isn't above the top of the slot after it settles, you have a valid sample that won't cause any problems with handling in the lab. We're using a heat and activation protocol, so the tubes are never opened while they have live SARS-CoV-2 in them on campus. The tubes come in, they're heat and activated in a scientific oven, and then after that you basically add proteinase K and do an RNA extraction free protocol. So this is the Saliva Direct protocol that takes you straight from the specimen to a qPCR test. And we managed to get that down to just 1 liquid transfer between those tubes and the 384 well qPCR plate. Then the assay we're running on that is the Thermo one. So that just covers the N1 and the N2 genes and does not have the RNA-seq control. By the time we're doing this in the fall, we will be using a new assay that also adds the RNA-seq internal control, and additionally adds, I think, flu A and flu B. So the idea is we should be able to simultaneously test all samples for COVID and for flu for the same cost that we're currently doing it with COVID. You might also have seen in the news the UCSD contract with county to do a pilot testing the schools. So that's led by Dr. Becca Fielding-Miller in the School of Public Health, and the Excite Lab is a part of that. And so what we're doing is we're trying to do the same sort of integrated framework in a way that's acceptable to schools, starting with schools that are chosen for being in low-income areas and without other testing strategies. And so this is very much an implementation science project, where the idea is to figure out things like how do you get a classroom of 6-year-olds to use those funnels without giggling catastrophically and getting their saliva everywhere, and other issues that are probably well known to those of you who have children. So, so that's about it for my update. I think we can go to the Q&A now, right? Thanks again.
So thank you so much. And thanks to all the speakers for their fabulous talks. We are exactly on time, so we have a great amount of time to go through our Q&A and the questions that were submitted.
Can I just ask a question?
Yes, please, Rob.
Yeah, so we do have sensitivity data on saliva direct. So if you skip the RNA extraction, your sensitivity is about in the 1 to 5,000 copies per milliliter range, and that contrasts with the extraction-based protocols, where you can get down to about 500 copies. So on the one hand, it's less sensitive. On the other hand, it's a lot faster and a lot cheaper, and we can do it at scale. One thing I forgot to mention. So a large part of the mandate of Excite is to rapidly test new technologies across UCSD that could be useful for improving COVID testing, and then get them get them EUA in a clear environment. So so we're already working with many faculty in the Jacobs School, in SIO, in the School of Medicine, obviously, in biological sciences. So, for example, we're collaborating with Omar Akbari on his LAMP assay, which we so what we can provide is real clinical samples that you can test on, and well understood assays that you can bridge to. And our explicit intention is to take all of the scientific expertise that UCSD has to bear on solving this critical problem for society. So please join us in that.
That's great, thank you. And that is really one impressive thing about the plan, is just the caliber of the individuals who are running it and the innovation that I'm seeing in all quarters. So thank you again. So I will start the Q&A. There were a couple of questions both that were submitted ahead of time and also in the chat about student compliance and how the plan really relies upon this. And so I was wondering if Dr. Scioscia would like to tell us how student compliance with the mandatory testing will be monitored, and what will happen if students don't comply with any of the policies.
Sure. And we've been thinking how to do this, and we're very fortunate that all of our students are automatically enrolled in the health system's EPIC process. So all our students have My Student Charts, and we get their results. And even the Excite labs will be through the EPIC process. So we'll have knowledge of when students are testing. We'll know their results as well, and we can time it all. So we're going to create a registry of all the students who are coming onto campus for a variety of reasons. They live here, they're on an athletic team, they're in a class, they're doing research. We'll put together that registry, and we can bounce that off of the EPIC data. So we'll be able actually to tell those students, send them a signal in My Chart, hey, you're due for your test. It's been 10 days since your last test. The other thing is, okay, day 17, you're out of compliance. We'll be able to create a list. And that will be going and working with the VCSA's office, because they deal with student conduct and student behaviors. The first step will be to take advantage of health ambassadors and other Triton health ambassadors. Give a call. Do you realize you're out? Is there a reason why you're off? Is there a reason why you haven't tested? If someone is consistently non-compliant, they will then go through a progressive disciplinary process. First, we give them a chance to be right. We educate them. We hope that we don't go that way. There's also the concern of, what if someone is in isolation and violates? What if someone is egregiously putting others at risk? They know they have the virus. They throw a party. That will be treated with very serious, including up to suspension and removal at that, certainly for the quarter. So Dr. Sutherland has been very clear, our BCSA, in talking to the students, that she will suspend anyone who puts others intentionally at risk. Now, we're hoping most of this is education and behavior. There's going to be a lot to make sure students have their masks. We're working on all those things to make it easy for them to comply. So I think that's the core of the mandatory compliance process for the testing.
great. Thank you. Okay, Quinn.
Dr. Scioscia, just a follow-up question. There was a question in the chat. Can and should students be asked to show a negative result from within the past 2 weeks before actually being allowed to enter an in-person classroom?
There's been a lot of discussion about this, and we actually have the ability to put on your screening tool that you're COVID compliant and that you're influenza compliant through some signaling. There is not an agreement that students will be forced to show instructors or will be asking instructors to show. On the other hand, if an appropriate person such as the VCSA and others authorized to check for it ask you to show evidence of compliance, you can do it through your daily screener app. You could actually intentionally open your app on your MyChart and you can show a test result. We're trying to avoid students having to show their individual personal health data. That's why we want to put a symbol on your daily screening thing. But the intent is not to have students have to have COVID monitoring checked every time they walk into a room or into their residential building. We're going to use this behind the scenes tool data, merging the EPIC data with our student registry and use that process rather than every day someone checking that you're compliant.
Thank you. And Dr. Anderson, there's a question about our, what is our contact tracing capacity and how much experience do the tracers have currently?
Yeah, so we've had a contact tracing team on the campus for about 4 months now. And let's see, yes, making sure that's right. And we have the capacity to quite comfortably handle 30 cases and 120 contacts per day. We have created a scenario that allows us to prepare for a surge, whether that surge is related to bringing people back from across the country, for which community transmission rates are very different and they're on our campus and we need to take care of them, to having outbreaks on the campus that may be larger than we anticipate. And in that case, we've created a force of ready responders. We have a reserve team that we would call upon to manage during those times that are much greater than what we have been seeing typically.
That's great. Scott, do you have a question?
Yeah, so this was a question in the submitted. So UCSD's COVID dashboard is one of the more meager ones out there compared to, say, University of Arizona or Bucknell's, for example. Since this can be a fast-moving public health problem, are there plans to increase the update frequency? The last data right now is from 10 days ago. The amount of information, the detail that's presented on that dashboard? And I'm not entirely sure who that should go to, but if any of you have expertise in that?
Yeah, that's very much a work in progress. Jennifer Holland has been the lead on this, working with Matt Hale, who populated the health dashboard, which is really quite strong. One of the things that we'll be doing is not just building a dashboard that shows cases, it'll be showing a lot of the data you heard about today from multiple streams, including behavior, capacity. It will be quite robust. We will use it for a lot of it for our internal monitoring of activity and trends on the campus, as Dr. Martin talked about. And then the key data about numbers of cases and so forth will be on the outward-facing web, harmonized in a way that you can compare it to county data, so that we'll have the same number of days of rolling average and that sort of thing. People won't be comparing apples and oranges. We've been building a boat as we've been sailing it. You've heard how rapidly Rob's lab has been coming together. You've heard about the ongoing refinement of the modeling. You've heard about the development of the contact team. You've heard about, we haven't heard today about all the isolation beds. All this has been happening at one time, and we need to get all these things together to come together with a data depiction that actually is adequate to reflect all of the stuff that's going on behind the scenes. But it will be there, and I think you'll be quite happy with what you see.
Okay, that's great. Thank you. I think many of us have been getting this nice PDF from the health system that has lots of daily testing data. I don't know if all the faculty are getting that or not, but that's a really nice document. Okay, so there was a interesting question about lessons learned from other campuses and how the modeling applies. I know this was answered in the chat, but I was wondering if either Dr. Martin or Dr. Goyal had anything else they'd like to say about what we can learn from other campuses.
Yeah, thanks. I'm happy to expand on that. I mean, everyone's been really interested in, of course, reading all the newspaper articles about what's going on in Illinois and Arizona and, you know, SDSU. I mean, I think Illinois was a unfortunate example of, you know, where, you know, you can test as much as you want, but if there are people that are knowingly violating isolation and going to parties positive, then that undermines that approach. And in a way, I feel like that supports our general approach, which, as you've been hearing today, is really this multi-pillar approach that's supportive of each other, and also we have redundancies within each pillar. So we're not relying just on one thing, such as testing. We have our risk mitigation strategies plus our viral detection. Testing is part of that. The wastewater monitoring is part of that, and the public health intervention. So again, you know, I think that one of the things that we did learn from Illinois is that I think a multi-pronged approach is necessary. And again, the ability to pivot or change approach when you see that something isn't working. If you see that if we see that there's an area where we're weaker, if we see that kind of behavior, or if we see that, that there's an area where maybe we have less uptake in testing among a specific population, or clusters of activity in that specific population, then we can adapt our approach, and we can also adapt the model and try to understand, do we need to increase testing to compensate for this? Do we need to boost our reserve contact tracers because we're sort of falling short on this? So again, we'll both be kind of looking at the individual metrics to see what we can do to support them and improve them, but also looking at how we can support this holistically across the board to really have this integrated strategy. And then, yeah, there was a question about benchmarking. I mean, it's a difficult thing. We want to do that, right? The aim is to collect data, try to learn as we go. I think the hard thing about benchmarking to campuses that we reopened in a rigorous way is the lack of ability, you know, is the lack of an analysis that has done that comprehensively without bias, trying to understand what strategies campuses have done, what they have not done, and then the kind of outcomes. Of course, we get all the reports of the Illinois, for example. But I think we are, you know, trying to learn some basic principles, and one of them is that, you know, off-campus behavior and parties is a likely substantial source of infection. And it's interesting because you've seen this pivot in some of the universities where they see these outbreaks off campus, and so they shut down all in-person instruction. And I think there's kind of a disconnect here, and what we're trying to do is, again, try to tailor our interventions to where we see risk and where we see the most risk of that transmission kind of furthering and occurring on campus. So really learning from those types of things as we go. But honestly, you know, in terms of behavior, my focus is on collecting the behavioral data, as much of it that we can, both the sort of digital proximity data that we'll be collecting through but also surveys, and I'm trying to understand adherence to masking, compliance with social distancing. And again, as we accumulate those data over time, we'll be feeding that into the models and revising as we go.
So if I could just follow up, there's a question about the masking and the social distancing, which was, do we have information yet from other campuses about the adherence to those two crucial pieces of the puzzle? How? Yeah.
Yeah, so I mean, I think the critical piece that we're missing is really, there have been some studies that have looked at kind of the proportion of time that students mask, and how, you know, that might vary by particular student characteristic at some. I've seen some analyses at isolated universities, but they haven't gotten into the nuance of when masking occurring off campus in these parties, for example, and who is it that's masking. And it may be that there is a very small proportion of individuals that are in these very high risk circumstances interacting without masks, and that's really where you see the viral transmission. So I think we need to be looking at those data and monitoring those in a more kind of nuanced way, both across, what do we see across campuses students are walking around, but also in these high risk situations. There was an associated question, slightly associated, that was asking about campus interactions and how we account for them, that maybe I'll take this moment to answer if that's okay. So we, as I mentioned, we have students interacting inside classrooms. They're allocated to classes based on actual registration data, as well as inside their residential facilities. But we have what is this, it's not a fixed number, there's turnover every day of this campus interaction network. And it's basically just assuming that there's random interaction with everybody who's interacting due to general campus interactions. So we don't simulate exactly movement from students from one classroom to another, but we assume, we kind of account for that outside classroom interaction in our general campus interactions.
That's great.
So this is another question along the lines of some that's submitted, and also one of my own questions is, given that yesterday San Diego saw that our numbers jumped from 6.9 to 7.9, and a lot of that came from the spike from SDSU, and you know, I understand that there's going to be another week before we really determine if we go back to purple. Do you foresee that that change will affect your plans at all, whether it be modeling or contact tracing, or how often we test, just so that we can kind of try to bring those numbers down again? I'm not sure who that question should go to, but.
Maybe I'll start from the modeling side, and then others can chime in. So from the modeling side, as I mentioned, we've actually developed a separate suite of simulation models that are used for county level forecasting, not just for San Diego, but other California counties as well. These are online. In the slide I mentioned is the link. You're welcome to go for it, go to see it. Those models use daily hospitalization as well as intensive care bed data, as well as cumulative mortality data, as calibration data to try to understand and infer transmission in the population. They're age stratified models, and we are using them to both forecast what's going on in the community and understand that community transmission, as well as I mentioned, feed them into our models to understand the impact of changing external incidents to what we might see on campus. So in terms of the changes that we see in the community, if we see increases in, for example, COVID hospitalizations and associated things, then that model would say, okay, we're seeing increased level of transmission in the community, and then we would then feed that into our model. The county level model also incorporates changes in public health interventions. So for example, we can use that model to simulate, okay, we can see that businesses they're set to reopen, or bars or restaurants are set to reopen. The last time we saw this change, what might we expect moving forward? So they actually allow us to kind of forecast a little bit and be maybe potentially one step ahead. And then again, we plan to feed all that information into the model to try to see how that could affect importation of infections on the campus. And then again, if we see the community incidence rises and is increasing, and then we look at our model and we see that affecting transmission on campus, then maybe that's a period where we increase our testing, or, you know, again, adjust our approach based on what we're monitoring. We're not just monitoring what's going on in UCSD, we're also looking at what's happening in the county, because we sit within the wider community. We're interacting, you know, with the community. We have faculty and staff and students living in the community, so we are accounting for that.
Yeah, I think in addition to what Dr. Martin said about, you know, adjusting the models and looking at the models to inform subsequent action, we can also, you know, sort of just look at what's happening in the people. And we've already seen over the last 5 months that the times when our campus has had more student cases or more employee cases have been times when community transmission has been higher in San Diego County. The other thing that we have the benefit of is insight from some of these other campuses that have opened before we have. And that insight sends us a message of the higher risk and vulnerability of our off-campus students, whose behaviors may, you know, put us in a situation of heightened infections and transmission. And so we've had some discussions about how do we take care of our off-campus students so that ultimately there's that collateral benefit to our greater San Diego community. And this could happen, you know, along the testing, tracing, and treatment portfolio, in that as we have our off-campus students back, making sure that it is easy for them to test, whether, you know, they feel like they've had an indiscretion and they need it, or if they're feeling symptomatic. We also want to make sure that when they do test, if the test comes back positive, we have the ability to get to them and their close contacts as soon as possible. If we notice that there's an outbreak potentially budding, and we have ways of monitoring possible outbreaks, we have teams that are set aside specifically for that, then we would work with the San Diego County outbreak team to jump on that outbreak prevention as soon as possible. And then, you know, we know that our students often, when they live off campus, they live in housing scenarios that aren't where you and I, you know, may currently live, multiple students to a room, making it very difficult to do some of the interventions that we need for isolation or quarantining to prevent transmission. And so allowing our on-campus facilities to be available to the students who need a safe place to isolate, a safe place to quarantine, is something else that we could adjust according to how things are playing out on the county level.
That's great. Thank you. Does anyone else have anything to add to that conversation? Okay. So I had a question about how might the wastewater testing impact the modeling? I'm wondering if Dr. Martin or Dr. Goyal have thought about that, and how much further that might depress the rate of transmission.
Yeah, so I mean, I think as we saw with what happened over Labor Day weekend, wastewater testing does have the ability to support our viral detection pillar by detecting outbreaks early and potentially detecting, like we saw in that incident, you know, 2 asymptomatic employees. And so really capturing people with asymptomatic infection to prevent them from transmitting to others, and potentially detecting clusters of transmissions at a very early stage before it escalates into a larger outbreak. So I am very excited to see where that goes. And again, as we collect data on the wastewater testing, we'll be trying to understand these relationships between viral positivity in a residence or cluster of residences, and what that signal looks like in the wastewater, how long that signal persists for, whether we can understand something about the relationships between the quantitative value of virus in the wastewater sample and potentially the number of people infected in the dorm. Those are things that we don't understand yet, that we are trying to understand as we move forward, so that we can better both kind of understand the utility of wastewater testing, but then also feed that subsequently in the modeling to say what kind of benefit that would have if we were to expand that. Rob, is there anything you wanted to add to that?
No, I don't think so. I mean, it's a wonderful opportunity to work together across disciplinary boundaries, and I know there's a lot of biological sciences faculty who are experts in things like population modeling, in viral evolution, viral dynamics, host pathogen dynamics, and so on. I think would certainly be extending the invitation to this community if anyone has techniques that they think would be exciting to incorporate on that front, as well as on the lab procedures. We'd certainly welcome that input. And I should also say the wastewater testing is going to involve a huge army of student volunteers, at least as currently perceived. So the risk is very low, the procedure is simple, the PPE requirements are very low. If you have students who want to get directly involved in combating the COVID pandemic and perhaps subsequently looking for letters for med school and that kind of thing, this could be a really good opportunity, and we'll keep you posted on those opportunities as well. But I think there's a lot of reasons why we should come together as a whole campus to solve these problems rather than having it compartmentalized.
Thank you very much. That's a great point. Okay, Quinn, do you have a question?
Yes, I do. I actually had another question for Dr. Martin. Specifically, how does the model account or does the model count for a degree of non-compliance or I guess people, yeah, so non-compliance is probably the best way to put it.
So do you mean non-compliance with what, is there a particular area that you're interested in, or just non-compliance with any of the activities on campus?
I think mainly a lot of the concerns could be with masking and social distancing that students may not be so great at doing.
Yeah. So again, this is a great question. It's the big, one of the big question marks. What I showed you today were simulations that assumed that there wasn't any masking and social distancing, and what happens if there is masking and social distancing and it reduces transmission by half, for example. Now, one could imagine that if people were willing to remain masked at all times, and were very assiduous about that, wearing their mask correctly and, wearing it, inside of their suites even, as well as rigorously adhering to social distancing, then you might expect to see a greater reduction in transmission than just the 50% we simulated. So, there is really uncertainty in that, but we've shown you the sort of 0 to 50% reduction in transmission, and it, what we see could be between those, or even, less transmission than we predict if the uptake is higher. So again, I think this goes back to my first point in terms of the aim is to collect data to reduce this uncertainty. We'll be collecting data as we go, and then we'll be refining those projections and sort of, updating that as we go, as well as trying to understand how we can increase the messaging to the students, provide them support, have student ambassadors connecting with them to really understand what are the barriers among students who are unwilling to mask or are participating in activities that may be risky, and how do we address them, and how do we engender this community participation and support for public health and everyone, support for reopening campus activities.
Yeah. And since you had mentioned ambassadors, I wanted to give Dr. Scioscia an opportunity to talk about how the student Triton Health ambassadors will be actually used to, I guess, help increase compliance on campus.
Yeah, the student health ambassadors, they will have 150 to maybe 250 students. That'll be a bit of paid stipend to do this. They'll be out educating, handing out masks. We see them also helping with the self-administered testing, which we've actually moved to, and being supportive of those efforts. I think they're part of the culture of students themselves driving this behavior and peer engagement in the process. I think they're going to be leaders for that peer engagement. They'll have logos and t-shirts and some other things as well, and some tools, but they'll have supplies to help students. They'll have educational materials for students as well. And so we really believe that the students being owning and being engaged in this process is really critical. It can't be administration only saying students do this. It's got to be students wanting this to work, students helping each other stay safe. We're calling it Tritons Care for Tritons. I think that's really important, that the student bodies, I think, are going to be an important part of this platform to be effective.
Thank you. That's great.
So there's a question about bad scenarios. So what are the thresholds for deciding that this isn't working? And what are the plans in place for in that eventuality?
That's an interesting question. I suppose to go back to what we were saying in terms of monitoring. So we have a number of metrics that we'll be monitoring on a daily basis that will be showing us within these areas. Is there a concerning trend? Is there something where we're doing well and that's fine and we don't need to worry about that? How's our isolation housing? Are we well within capacity, or are we nearing that, right? So on a number of metrics, both within our testing, our contact tracing, community level epidemiology, wastewater testing, symptom screening, some modeling in terms of the reproduction number on campus, may have for behavioral metrics. So all of those things we're going to be kind of monitoring, and hopefully we'll see that most of the time we're doing well. Maybe we'll identify that there's a cause of concern, and that's when we'll discuss, adjustments of action. But again, this is going to be an ongoing daily process where we, are monitoring what we're doing and trying to assess whether we need to change course or whether what we're doing is working. And we can, and we can stay with the approach that we currently have. Chip, do you want to add to that?
I was going to also comment in the other direction. There was an interesting question from Dan Hines about, we hear about all the disasters. Are there places where things are going well? There actually are quite a few places that are going well. There aren't a lot of reporters who hang out and breathlessly report that things are going well here so far. But I've been in talk. I'm in touch with a number of places. I've talked to my counterpart at Duke. Things are going very well at Duke. Things at Berkeley so far have gone well after they had that pre-summer activity. Every Thursday morning from 11 to 12, I get on the phone with counterparts in all the UCs, and the places that have opened so far, Merced and Berkeley being on semester systems, as they've gotten into things, they're kind of in the so far so good situation. Case Western Reserve is doing well. So I think there are examples of things that are going well. EVC Simmons is constantly in touch with her counterparts. There was kind of a national call of all the EVC and EVC level positions earlier this week. And the campuses talked about things that were going well and not going well. And she said the places that were going well, when they talked about what was working, what they mentioned were always the things that we're doing here. It was everything was mentioned were things we have as part of our kind of boringly redundant pillars program. And so we're hoping that the successes that have been seen in other places we can emulate here as things ramp up. But your question's a very good one. We need to learn from both the successes and the failures, as with most things in life.
Just to follow up, I think it would be great if this works and we don't see an outbreak. I guess the question is, have decisions been made about when we decide that the outbreak is too big? How we do in that case? The worst case scenario, has that been discussed, I guess, and what are the plans for that?
I think we're also approaching this is that there are a lot of iterative steps before you get to worst case. And we would be remiss if we didn't start actively putting the iterative steps in early and quickly. And that's where we're coming up with this daily monitoring, a core group that's looking at this stuff every day, a series of iterative steps and interventions that can be done in advance to bring things under control. So I think that, yes, we could get there, but there's a lot of runway and a lot of opportunity for interventions along the way to avoid a complete, everything has to go remote, everybody has to go home, or we have to shelter in place. And so we call them public health interventions. Dr. Anderson has taught us well the terminology, and it's great. There are a lot of things we can do along the way that slow this down. And if we communicate effectively and are signaling, and that's, I think, where all of our testing capacity is so great, we actually get signals very, very quickly if something's going on. And our contact tracing and case investigation is strong and deep. We've been doing it with the students. We've got the public health teams. We're all integrated on doing this. So I think we're going to be able to reach out quickly and bring things, hopefully. There will be a few things that will happen. We know that there will be some virus that will get into some students and faculty and folks. We've had employees. It's going to happen. But we can manage it and control it in a way that it doesn't cause us to stop.
And one of the things that I think we are trying not to do is what a lot of the public health models have been, here are the thresholds at which we will do X. And one of the issues that leaves you doing is saying, well, we don't need to do anything until we get to 6.9 cases per 100,000. You can see it going off the rails a week before that. The supervisors have done that twice already, waiting until we're not to the threshold yet, we're not to the threshold yet. And then when you get to the threshold, you've got all that momentum that's built up in the past. It takes you that much longer to get things under control. So our goal is to be looking at trends and trajectories and to be responding as we go, rather than setting arbitrary thresholds that we don't even want to get near. So it really is an issue of proactive management rather than reaction to thresholds that have been crossed.
Okay, thank you. So it's now 6:31, and I just wanted to ask our guests if there's anything that they would like to say in closing that hasn't been said in the Q&A session.
Well, I'd like to thank everybody for being here. It's helped us a lot think about what we're trying to do, and I think your place on the front lines as we get people back will help us tweak what we do. So we would appreciate input as we go, and things that you see and opportunities that you identify, we're all ears. Rob and I and are both faculty members where, so we want this to work, and we need your help to have it happen. And we've tried to bring the most creative people to the table, and I hope you've seen that today. Natasha is also a faculty member, didn't mean to overlook her, but my point is this is really a faculty-driven process that we've taken to the administration. And we've been lucky because they've essentially said, what do you need to have this happen? We've been very fortunate with the Chancellor saying, and his Chief Financial Officer, when we've identified a need, it hasn't been, well, can you get by with X? It's been, let's get that done. We were assigned very early on essentially a Chief Operating Officer, Bob Newhart, who's incredibly talented, and he's like a bull going through cement walls when something needs to get done. Without that, all these ideas you've heard of would still be just ideas we'd be talking about and complaining that the administration wouldn't let us implement. So Rob Knight's lab would have taken forever to get going in a lot of other scenarios when you don't have a chancellor willing to write the check to buy advanced liquid handling equipment. So I think we're fortunate to have a lot of people who are thinking a lot about this and an administration that wants this to be successful. And so if you see ideas that you think we can implement to make things better, I think you'll see that there is receptivity at the part of the people who can provide the resources to get it to happen.
That's wonderful. That seems like that's really great to hear. And I think that's probably an appropriate place for us to stop. And I just want to, again, thank everyone who came to join us this afternoon. Thank you so much for your leadership in this amazing crisis that we're following. This really does, to me, illustrate the best of UCSD. We have people coming together across disciplinary boundaries and working together to help build the most robust plan possible. So thank you all for displaying this fabulous teamwork and innovation at the very highest levels. And thank you so much for your time. So thanks, everybody.
Bye-bye. Have a good night.
Thank you.